Hi everyone, so today I will be talking about the Vesicular Stomatitis Virus. All vesicular viruses possess a single-stranded negative sense RNA genome. VSV has 5 structural proteins: the nucleoprotein, denoted by N, phosphoprotein by P, matrix protein M, glycoprotein G, and RNA-dependent RNA polymerase, denoted by L. The genome itself is flanked by the 3' and 5' end, and has signals for making proteins and copies of itself. The viral genome is coated and protected by the M protein, and the P protein is joined with the L protein to make a complex with the N protein, and it condenses into a helical structure by the interactions with the M protein. This whole structure is then surrounded by a membrane taken from the host cell and is embedded with the G protein. The glycoprotein helps the virus stick to a receptor called the low-density lipoprotein receptor, LDLR, and this allows the virus to infect many different types of cells because this receptor is found in many different cells. After sticking to a cell, the virus gets pulled into the cell in a process called the endocytosis. Inside the cell, the virus travels through a pathway called the endocytic pathway. This pathway makes the environment more acidic, which causes a change in the shape of the G protein on the virus's surface. And this change allows a part of the virus called the fusion loop to stick out and merge with the cell's membrane. When the two membranes join together, the virus's genetic material is released into the cell. At this point, another part of the virus called the M protein separates from the genetic material, allowing the virus to start making copies of itself. Positive or negative sense RNA virus refers to the polarity of the RNA genome, and the polarity tells us whether the RNA can be translated directly by the host cell's machinery. If it's a positive sense virus, it can be translated directly. But VSV is a negative sense virus, meaning that they carry within a RNA-dependent RNA polymerase to transcribe the negative sense RNA to positive sense RNA, which can then be translated by the host cell to make viral material. The polymerase moves in one direction, starting from the 3' end and moving towards the 5' end. It transcribes the genes one after the other in a specific order: N, P, M, G, and L proteins. Sometimes the polymerase stops at each section, so the closer a protein is to the 5' end, the less of that protein is made. Once the virions are assembled, it ultimately buds from the plasma membrane with the most G protein. This entire cycle kills the host cell quickly. The vesicular stomatitis mostly affects the oral mucosae and the feet of the hoof animals. I won't show photos to keep this video PG. Otherwise, that's all for today. Thank you for watching.